Hello everyone and welcome to this ELISA webinar. I think before we get started with the practical stuff and the presentation itself, I think we'll just wait a few minutes to get everyone into the live webinar. So hold on tight. In the meantime, Ivan, I think we could just talk about what you think sets cell and gene therapy apart from other biologics like recombinant proteins or vaccines and monoclonal antibodies. What's different here?
I think that's a very good question. So I think the first issue is that these products are much more complex. So they are more difficult to manufacture and they are more difficult to control. So your analytical methods need to be more robust. They need to be able to measure many different analytes. So that's issue number one. The second issue is that the technologies for making these products are improving a lot, very fast. So that means that the analytical methods you apply also needs to be flexible. For classical biologics, the manufacturing process has been optimized for many years, for example, for monoclonal antibodies. And the analytical methods have been optimized and fine-tuned for many years. So this is a brand new area of exciting therapeutics and development of better manufacturing methods and improved analytics.
Yes, thank you. I completely agree. It's a really interesting topic and really different from the other biologics as well. So I think I see some people coming in. So I think we'll get started with the practical stuff so you can have the presentation. First of all, my name is Martin Rask Johansen. I work as a protein analysis advisor at ELISA, and I'll be your host today. For this topic today about analyzing process-related impurities and proteins in general in cell and gene therapies, we have brought in our expert on the field, Ivan Mertz. He holds a PhD in mass-spectrometry and analytics for biologics, and he has been working with protein analysis for more than 20 years in the industry, and he's also the co-founder of ELISA. So for this webinar, Ivan will present for around 20 minutes, and let's keep it at that. And then afterwards, we'll have around 10 minutes for questions. So if you have any questions for this topic and for the things Ivan presents, please put it in the chat. Also in the chat, you can put in comments for the technical part, so our colleague Jette will know if there's anything wrong with the image or the sound quality. So I think on that note, we'll just get started with the webinar. So go ahead, Ivan.
Well, thank you very much, Martin, and welcome to you all. So I'm really happy to see that so many have signed up for this webinar. So what we will talk about today is how to ensure consistent manufacturing and analytics for cell and gene therapies. So we will cover three topics. So the first one is how is FDA requesting more consistency to the manufacturing and analytical methods applied for cell and gene therapies. And number two, what is the complexity of these samples that makes it so difficult to use classical impurity analysis methods. And number three, I will go through mass spectrometry methods. It's a method with one single method, you can identify and quantify all different proteins in these complex products. I have three case examples from different gene therapies that I will also show to give you some inspiration how the methods can be used. So with that, I will just go to the first slide here. A little bit of background about ELISA. So we are a contract analysis service company located here in Denmark. We also have an affiliate or a company in Palo Alto, California. So we have a lot of clients throughout Europe and North America and a little bit in the rest of the world. So we provide protein analysis services to help biologics development. So we have specialized in classical biologics, proteins, peptides, maps, vaccines, biosimilars, and also the past 3 or 4 years, a lot of gene therapies. The analytics we are doing is analysis of process-related impurities, HCPs, and also viral proteins in cell and gene therapy products, as you will hear about today. So we have different analytics for these impurities listed here. We also do classical characterization of the drug proteins itself. So these are typically peptide mapping, intact mass analysis, quantification methods, and different analysis for force degradation studies that I will not come into today. So the first part of my talk is actually illustrated by this publication earlier from this year, titled FDA Seeking More Consistency from Cell and Gene Therapies Developers. It's a publication, you can read more details on this link here. It says the FDA is pressing cell and gene therapy developers to more consistently measure the characteristics of their complex products. And it says that several products have been delayed by FDA to improve consistency and improve the test methods used by these companies. So this is the topic we want to address. So first, there are also guidelines that are very recent. So one from January 2020 from FDA, where you can read a lot of the CMC details about it. There's also one from EMA with a similar title from 2018. So not to go through all these documents, just an example on the impurity chapter from the FDA guideline. We recommend that your manufacturing process be designed to remove process and product-related impurities, and that you have tests in place to measure the level of these impurities. And then at the bottom, list different types of impurities, including viral proteins and reagents containing other proteins. So clearly there are some good guidelines how to do this, but it doesn't say exactly what methods to use, because these methods are not always present or often not in place. So why is it so difficult to measure these type of products? One of the reasons is that they are very complex. The samples are very complex. It's a mixture of different proteins from different sources. I've listed some of the sources up here. So one is the viral vectors used. So each of them contain a number of different proteins that are part of the product. It also has a production and packing cell line. So typically insect cells, ASF9 cells, different types of human cell lines like the HEC293 cells, but there are also more rare cell lines used by some customers, for example, murine cell lines. And then we have the manufacturing system and the growth medium and reagents used during the manufacturing. So typically we see bovine serum used in the growth medium, human albumin, antibody columns, enzymes added during the manufacturing. That also complicates the samples. So it is a mixture of many different proteins and many different components that can influence with the analysis. So this makes these complex products very difficult to measure with classical protein analysis methods and classical impurity assays. For example, ELISA is only developed, as you know, for one species of protein. It cannot measure all across different species. So what can we do about it? So here at AlphaLife, we have developed mass spectrometry methods for many years, and we have developed a mass spectrometry assay based on the technique called SWOT LCMS that can both identify each protein, and it can also quantify the proteins in a very reproducible way. So I'll just take you through the principles of this method and then show you some applications. So the first step in this procedure, excuse me, I'll just take a little water. So the first step in the analysis is that we spike in some known amount of intact protein. We use these as internal standards in the sample, and we use them to make calibration curves for the quantification. We also use them as quality control samples to make sure that all the sample preparation steps have worked as they should. Then we precipitate the proteins. We do this to remove all the different buffers from the different solvents the samples may be in. And then we cleave the proteins into peptides with an optimized digest procedure. And then we prepare the peptides for LC-MS analysis. So we don't analyze the intact proteins. We analyze peptides by the LC-MS instrument you have seen here on the photo. So here we use a microflow LC system that gives a very robust chromatography. And we use a high-end mass spectrometer from SciEx that can measure these low amount of proteins and impurities in very complex samples. The third step is to take the mass spec data and then identify the proteins. So to identify the proteins, we set up a database containing the protein sequences of all proteins that potentially could be in your samples. So this includes the sequences of all the viral proteins. It includes the proteins from the expression systems, and it includes also the proteins you may have added in your manufacturing process that potentially can end up in your product. Then we search the data against the database, and then we end up with a list of proteins as illustrated here. To make sure that all the proteins on the list are identified correctly and not just one-hit wonders, we say that we need to observe at least 2 peptides for each protein to be identified. To quantify it, we use the sum of the MS signal for all peptides belonging to a specific protein and then correlate it to the internal standards. So we end up with a list of proteins identified and the amount of each protein in the sample. So the first example I would like to show you is from adeno-associated virus. So it's a UK-based cell and gene therapy company. They're expressing the adeno-associated virus in SF9 cells using a baculovirus expression vector. And the objectives in this project was to compare the protein profiles of 6 different batches of drug substances from 2 different AAV products. So, and the question is the purity and protein profiles consistent between the batches? And they also asked us, what is the reproducibility of your method if we are going to send more samples later on for the same analysis? And the second objective, are there any specific host cell proteins that can be potentially problematic or of concern for product stability or eventually for patient safety? So both compared to what AlphaLISA has seen in other AAV products, but also what is known in the literature from classical biologics. So if we look at some of the data, so here we only look at the viral proteins resulting from the analysis. So what you can see is the total viral content in nanograms per ml for the 6 different batches. So we can see for product 1, the viral content is quite consistent between the 3 batches. For product 2, 2 of the batches were quite similar in the content, whereas the batch number 6 had a much higher content or concentration of the viral proteins. When we looked at how reproducible the method was, we did a triplicate analysis, and you can see in the bottom table that the coefficient of variation is very good. Typically, it's between 3 and 6% here, 7%. Normally, it's within 10%, we say. So this is much higher than what you can obtain, for example, with a standard ELISA assay. Then the second part of the analysis was to look at the host cell proteins detected in the AAV. So the information is, of course, confidential for this client product. So that's why it has been disguised for some of the proteins. So, but you can see the principles of the analysis. So you get a list of proteins, you get the amount, you get the theoretical isoelectric point and theoretical molecular weight of each of the protein. You get the names and the database accession number, so you can look up the protein in the original database and learn more about this protein. You can also evaluate it for potential unwanted properties in your product. So what I've just highlighted here are 4 different proteins that we often observe. So one of them is ubiquitin. In itself, it's not problematic for the patient, but ubiquitin can mark proteins for degradation. So it is a potential problem that if large amount of the drug is ubiquitin-related, then it might be partly degraded. So you want to monitor this protein. Also, there is a peptidyl isomerase, and this protein is not known from SF9 cells to be problematic, but from CHO cells in MAP product, it has been known to cause some product aggregation due to post-translation modifications of the MAP. So these proteins you may also want to monitor and perhaps optimize your process to remove this protein. Case example number 2 is from lentivirus. So here the case is that the virus is grown on HEC293 cells, and they are grown on a complex growth medium containing human serum albumin. So, and the objectives are again to quantify the different viral proteins from the lentivirus, determine the amount and ratio of different viral proteins. Also, this client asked us if we could look for the transgene. It's a T-cell receptor expressed in the humans, and they also ask if it's expressed in the HEC293 cells. And thirdly, what is the amount of residual proteins? So just looking at the data, if you look at the picture at the bottom of the figure, you can see the viral proteins. So where you have the GAG polyprotein and the GAG polyprotein. So the protein expression level from these two and the ratio between these proteins, they are critical for maturation of the virus and also critical for correct viral assembly. So as a manufacturer, you want to monitor that you have the right ratio of these proteins in your product. So, and as you can see the table above, we can quantify each of the viral proteins, and you can look at the amounts and compare them between different batches. We could also see that the T-cell receptor was expressed in small amounts for 3 of the products. On the table on the right-hand side, you see the total amount of viral proteins in nanograms per ml. So really, it gives a lot of details about the proteins in the product. We also looked at the host cell proteins from the HEC293 cells, and from the table here again, you can see a long list of different proteins in the different samples. We can also identify the proteins from the growth medium, and we knew that human serum albumin was added, so we identified this specifically. So that's the top protein on the list, so a lot of albumin in there, but we also see other human proteins in lower amounts. So you can use this to monitor your product for consistency. The third example is the adenovirus. And the adenovirus was expressed in also a human cell line, a different one. And the client already had ELISA assay on this cell line, but they measure inconsistent amount of the HCPs in different batches, typically between 10 nanogram per ml and 100 nanogram per ml. So the objective was, again, compare the HCP profiles between 3 GMP batches using the LCMF method. Are any difference between the batches? And then secondly, identify and quantify the viral proteins. Are all the viral proteins detected, and are the batches consistent in viral content? So here again, anonymized the list of HCPs. We found 7 HCPs. The total amount of HCP is summarized in the bottom of the table. So you see nanogram per ml, around 50 nanogram per ml. And we compare 3 different batches, and they look quite consistent, perhaps with a little lower concentration in batch number C. So we both get the individual viral concentration and the total viral protein concentration. Here we look at the viral proteins. So you can see we identified about 14 different viral proteins from the adenovirus, and we get amounts on each of them, and again the total amount. Again, you can see that for batch C the viral amount is a little bit lower, and that might explain why the host cell protein amount was also lower for this batch. The alternative method that this client was using is the classical 1D SDS page. You can see an image on the slide as well. So here you would need to run the samples on the SDS page, do sensitive silver staining to detect only some of the proteins, and if you wanted to identify them, you had to cut them out and do LC-MS protein identification. So that's a very long and tiresome process where you don't get actual concentration of each protein. So the new method has a lot of advantages compared to the old method. So with that, it's about 20 minutes, Martin. So I'll just summarize. On the LC-MS method, it has a number of advantages. So you can identify proteins from multiple species by the one and same method. You can both identify and quantify the individual and total proteins, both the viral proteins and the host cell proteins and other proteins you may use in your manufacturing process. We will check that all the standards perform as they should, and then maybe adjust a little bit to your specific metrics if there are some things that are not working as expected. But for now, we have analyzed more than 150 different projects and a lot of different products with the same general method. It only takes a few weeks to adjust it and apply it to a new type of product. Because it is so reproducible, we can also validate the method for your needs. We have ongoing projects for method validation and are currently taking the analysis into a GMP for potential release testing of specific products. So just to summarize the take-home messages from this webinar. So how to monitor impurities and how to ensure consistent cell and gene therapies. So we talked about how regulatory agencies request more consistency from developers of these products, particularly on the manufacturing process and the analytical methods. The high complexity products make it difficult to use existing methods, so everyone is looking for better methods. And with the mass spectrometry method, we have one method that can reproducibly quantify and identify all the different proteins from multiple sources in your samples. So with that, I hope I have given you some inspirations, and perhaps you have some questions. We can take them now if you write them in the chat, but you're also welcome to contact me by email or connect with me at LinkedIn. So I'll be happy to discuss and take questions from you. So with that, Martin.
Yes, I'll jump in again. So as Simon said, we're ready to see some of your questions. So please put them in the chat. I see we're already getting some, so that's great. But first of all, I mean, I think we could start us off by talking about the protein quantification, because it's a question we get a lot. How is the protein quantification obtained? Can you give us a bit more detail than you did?
Yes, I can. So I think that is actually one of the secrets for obtaining the reproducibility of our assay. So before we start any treatment of the samples, we spike in these internal protein standards in known amount. So these are intact proteins, and we know exactly the amount. So when we do the digestion and cleanup of the peptides and LC-MS analysis, we can generate very good calibration curves. So we know the ratio between the protein amount and the MS signal. And we use this to quantify the unknown proteins in the sample. So it becomes a very reproducible method, and it will normalize away any differences in results based on, for example, different matrices and different temperatures in the lab and small things in the procedure. Yeah.
Do you see it as a potential to maybe set up an MIM method for specific proteins of interest?
Yes. So we also have MIM methods. If you're familiar with mass spectrometry, so we can also, this method I showed here can identify and quantify a lot of proteins in a broad way. It's a discovery method, you can say, but we also have MIM method where you specifically can monitor specific peptides for specific proteins to obtain higher sensitivity and maybe a more robust analysis for GMP, for example.
Yes. Okay, thank you. Yes, we'll get started on some more questions. What are the criteria for successfully identifying a peptide? This is more challenging in a non-targeted approach. Is the parent peptide fragmented?
Yes, so the peptide is fragmented into peptide fragments. So the criteria for identifying a peptide is that we should have at least 6 peptide fragments observed. The mass accuracy should be good, of course, and then the peptide is given a kind of a score to evaluate this.
Yes. Yeah, I hope that's answer enough. How much cell culture harvest do you need to establish the process for HPP identification?
Yeah,
often question as well.
So if we have, if there is plenty amount available, we would like to have half a milligram of each sample or 1 milligram. If it's more limited, we can also do with less. So total protein, half a milligram, 100 micrograms, 1 milligram. Yeah, in that range. Okay,
great. What about impurities from proteins that are not identified, less than 2 peptides?
Yeah, so these can either be so-called one-hit wonders because they are noise. So when we do this triplicate analysis, we will filter away those proteins. Other proteins might be in so low amount that only 1 peptide is observed. So that doesn't give a reliable both identification and quantification. We can have them on an observation list. So if there are some of potential concern with only 1 peptide, or if you have seen it in a higher amount in early process sample, you can monitor this specifically. Normally we don't report it because we are not absolutely sure about the protein.
Great. What is the expected limit of quantification for viral proteins?
So these the data as I showed you here that's in the low nanogram per ml. So typically we see a limit of quantification in the low parts per million. If we set up an MIM method, we can go below 1 part per million. Yeah. So it is just as sensitive as an ELISA or more sensitive, and you will know specifically what analyte you're looking at.
Yes, exactly. Yes, and is it possible to follow the reduction of 1 ATP in the downstream process?
Yes. So we have actually many projects where we're doing this. So we will then get the different process samples from the early harvest of the proteins to the final purified drug substance. Then we can monitor the amount of the given protein through the different purification steps. If you want to optimize the purification to remove this ATP further, you can see what steps are working, what steps are not working, and maybe you can adjust the pH or a buffer composition a little bit to remove it. So it becomes a very intelligent, data-driven process to remove specific ATPs of concern.
Yeah, it definitely does. I remember a customer case where one customer got the data and removed 1 specific ATP. I think it was in only 14 days because they could just do it intelligently. So that's great. Then we have a little broader question. What is swath? What are its advantages compared to MS? So I'm thinking maybe we could talk data dependent versus data independent acquisition.
Yeah, that's a very good question. So swath is actually a name invented by the MS instrument vendor Sykes. So other people call it data independent acquisition. So it means that the mass spectrometer will not decide to only analyze the most abundant peptides at a time. It will simply fragment everything at a given time. So this has the advantage for ATP analysis, that also the low abundant peptides will be fragmented and identified. So even if you have large amount of your drug substance or your viral proteins, for example, you can still identify low level of ATPs in a reproducible way.
Yes. And one more question I see at least here. For 100 milligram per ml map monoclonal antibody, can you look at peptide mapping and ATP in one assay?
Yeah, I think that's also also a very good question. You can say that we have the data, we have collected the data. Normally, however, the amount of the map or the drug substance is too high. We are outside the linear range of the quantification. So normally we don't do the peptide mapping on the drug substance from this data set. We have another workflow optimized for peptide mapping, for quantitative peptide mapping, to look at modifications and quantify these and characterize product-related impurities. So in principle you can do it, but we have another workflow that's more optimal for this.
Great. Then we also touch a bit of product characterization in itself. I'm thinking we could maybe just take 1 or 2 questions more, but then we'll call it, so we stick close to the 30 minutes. I mean, I'm thinking often we get a question, especially for cell and gene therapy, around Higgs cells and ELISAs for Higgs cells. Are there any good Higgs ELISAs out there, or is there a way for a project or a client to find out if their ELISA is good enough to cover their process?
Yeah,
because it depends on the process and the product.
Yeah, so there are commercial ELISA kits out there for Higgs and also for some of the other expression systems. When you use an ELISA, you always have to show that it can detect the impurities in your product and impurities that could potentially end up in your product. So for an early process sample. So we have a very good method for this. We invented some 3 or 4 years ago. It's called ELISA MS, where we do the coverage analysis of the ELISA using mass spectrometry as a readout. So in some cases there can be found a good ELISA that can detect the cell line impurities. However, it will not detect the other impurities from other species.
Great. Then we also touch a bit on coverage analysis, which is also, of course, related to this, because some people have an ELISA that they can use, so it's why not find out how good it is. But you also touch a bit about going into the GMP using mass spec only as a release assay, because when you have these cell and gene therapy products, some of them use an uncommon expression system, but has anyone established it in GMP, and what do the regulatory authorities say about this?
Yeah, so there are a few big pharma companies who have validated their own mass spec method for ATP analysis. I'm not sure if they're using it for release testing or just for characterization. Here at AlphaLife, we get a lot of requests from customers where it's not possible to find a good ELISA assay. So or they don't have time to develop a good ELISA assay. So for these products, it's urgent for them to have another assay, and they ask us to take it into a GMP environment so they can use it as a release test. So we are in the process of doing this now, validating the methods, IQ, OQing the instrument with SciEx, and we have some 3 different projects. And I can say one of them is a COVID-19 vaccine project, of course. So it is urgent, and we expect to be there in the first quarter of next year.
Yes. So I think more news on that development. But I think we should call it here. A lot of great questions. If you have any additional questions after this, please write to Eivind or AlphaLife in general through our website, and we'll also follow up with you to find out how you like this webinar. So thank you so much for joining this. We're glad to see so many attendees, and have such great questions. So have a nice day and talk to you soon.
Bye-bye.
Bye.